Hey everybody! For anybody that missed this week's garden lesson, grades 3 through 5, we did a book reading as well as a worksheet. And so if you were not able to make it to that class session, I wanted to make sure that you could still have fun, get outside, and use the materials that are already in your garden kit. So if you missed the lesson, you're going to go ahead and you're going to get out your garden kit. Okay? Lafayette Garden class looks like this. You had lots of good stuff in there. You're going to go ahead and you're going to get out your bug jar. Okay, this is a fun little bug jar. It has a magnifying glass as a top, so things are going to look a little bit bigger when you look through them. Okay, you're going to get out that. You're going to get out your loupe or your magnifying glass. That is yours to explore with. And you are also going to go ahead and get out the printable for buzzing with questions. If you don't have any of it, that's fine. I will go ahead and walk you through how to substitute different things, but that is what you can get out of your kit if you have it. Then what we're going to do is we're going to read the book Buzzing with Questions, which is a great story about Charles Henry Turner. And for those of you that did do the garden lesson, we read about half the book. So if you want to listen to the full book, this video will have the full book reading on it. Okay, so let's get started. This is the book Buzzing with Questions, The Inquisitive Mind of Charles Henry Turner, and it was written by Janice Harrington and illustrated by Theodore Taylor III. And so this book is about Charles Henry Turner, and we can make a few inferences from the cover. So you can see the different things that are on the cover. Okay. Charles Henry Turner was one of the first African-American entomologists. So if you've got your detective cap on and you're trying to figure out what an entomologist is, you can probably make some deductions using the cover art on the book. He's studying lots of little mini beasts, right? So we've got slugs and snails, we've got butterflies and grasshoppers, we've got ladybugs, and we've got bees, and it looks like a little roly-poly. Okay? All right, so buzzing with questions. Questions that itched like mosquito bites, questions that tickled like spider webs, questions you just couldn't shoo away. Questions hopped through Charles Henry Turner's mind like grasshoppers. His brain buzzed with questions about plants and animals and bugs. His parents' home swarmed with books, but never enough books to answer all of his questions. Charles Henry Turner asked so many questions that his teacher urged him to go and find out. So Charles did. He read and studied and worked hard, and after he finished high school, he did what many people thought was impossible. Even though he was a janitor's son, even though most colleges didn't accept African-American students, even though it meant more hard work, Charles Henry Turner went to college. In his biology class, Charles met the magnetic young teacher, Clarence Herrick. Herrick's classes hummed with energy, students chatting, students examining the organs of small animals, and students staring one-eyed through the microscopes. On Friday afternoons, Herrick invited students to his laboratory for spirited talks about biology. He spread tablecloths over the long laboratory tables and set out sweet cakes and cups of tea, but Herrick worried about inviting Charles Henry Turner. He worried that the other students wouldn't want to drink tea with his only brown-skinned student. To Herrick's delight, the other students wanted Charles to join them. They liked the shy, quiet student who always earned high grades, the hard-working Charles Henry Turner. Charles was indefatigable, a classmate said. He spent hours peering through microscopes, planning experiments, gathering specimens, keeping records, drawing charts, and reading scientific papers in French and German. But whatever the language, he never stopped asking questions. One question led Charles to a small, eight-legged, eight-eyed, two-fanged creature, the spider. Charles wanted to know if spiders could learn or if they were only weaving machines that made the same web over and over. Charles searched for spider webs. He trudged through meadows, inspected stone walls, and scouted the sides of railroad tracks. He toppled wood piles, lingered over logs, and peeked into dusty corners. Charles found webs, lots of them. He even spotted double webs that looked, he wrote, as if the spider were trying to fish with two lines instead of one. All kinds of spiders and all kinds of webs caught Charles' attention, even a web between a windowsill and a wall. What would the spider do, Charles wondered, if he swept away its web? With a broom, Charles brushed away the web. Not knowing it was part of a science experiment, the unsuspecting spider rebuilt its web. Sweep away, rebuild, sweep away, rebuild. After losing its web for the 5th time, the spider gave up and wove a new home beneath the windowsill. But Charles didn't give up. He repeated his experiment with an arachnium, which is a spider jar. He filled the bottom of the jar with sand and pushed a post into the sand. Then he added a spider. He slipped a paper triangle into the jar and watched the spider. Later, he removed the paper triangle, replaced it with an L-shaped tube, and watched again. With each change, the spider rewove and reshaped its web. Charles concluded that instinct told the spiders to make their webs, but that each spider wove a web just right for its home. Charles called this intelligent action. Spiders were not just weaving machines. The indefatigable scientist then wondered about even smaller animals. In scummy ponds and weedy ditches, he searched for tiny crustaceans. He found seed shrimp, water fleas, and wheel animals. Through his microscope, he admired their small bodies. They were beautiful and translucent, and some looked like a nest of test tubes. Charles even discovered a new crustacean and named it Cyprus herricki after his friend and teacher Clarence L. Herrick. But Charles wouldn't stop asking questions. At 39, he returned to school. He wanted to learn. He wanted to ask big questions about another small creature, the ant. Charles wondered how ants found their way home. Did ants have a hidden power in their brains that pulled them to their nests? Did the sun guide them? Did they follow a trail of smells? Searching for answers over the next 5 years, the indefatigable scientist studied acrobat ants, big-headed ants, false honey ants, and odorous house ants, all kinds of ants. To tell one ant from another, Charles marked their abdomens with watercolors. He built an obstacle course with cardboard stages, cardboard rooms, and cardboard ramps. Ants crawled over the stages and scurried over the ramps. Ants tapped the cardboard with their antennas. Ants searched for their nests, but Charles didn't help them. Instead, he moved the ramps to unexpected places. He angled ramps up and slanted them down. He painted ramps different colors. He shifted the light and the direction of the light. He smeared stinky oils all over the cardboard paths. He caused the ants trouble. But by watching ants find new ways to reach their nests, Charles learned how they solved problems. Charles drew a map with squiggly lines and arrows to show the wandering path of a lost ant. A French scientist called this wandering Tournement de Turner, or Turner's circling, in honor of Charles Henry Turner, the scientist who taught us that ants were not mindless robots. Each ant used sight, sound, touch, smell, and the movement of its body and sunlight to find its way home. Like the ants, Charles now searched for a new home. He found work as a biology teacher at the first African-American high school west of the Mississippi, Sumner High School in St. Louis. Excited by their new teacher, students filled their lab books with notes about his class. One student wrote that Charles set out dishes of jam for bees at breakfast, lunch, and dinner. The bees circled and buzzed at each meal. But when Charles set out jam only at breakfast, the bees still invited themselves to lunch and dinner. Surprised, the students learned that even bees sense time. Questions kept circling and buzzing in Charles's mind. In a St. Louis park, the scientist placed red cardboard circles in a patch of clover that swarmed with bees. He coated the circles with honey, and then he waited. The bees weren't interested in the circles. They weren't even drawn to the smell of honey. But good scientists are patient, so Charles tried again. He caught the bees in a bottle and toppled them over the honey. At first the bees buzzed away, but eventually they settled on the cardboard to enjoy a sip of honey. Once the bees learned that the red circles carried honey, Charles replaced the red circles with honey-filled blue circles. The bees ignored them. Whether he used circles or cones or boxes or added new colors, the bees kept flying to the color red, even when another color held lots of honey. In 32 different experiments, no matter how Charles tried to trick them, the bees chose the color red. Charles Henry Turner was the first biologist to prove scientifically that bees could see color. Bees, giant water bugs, whirligig beetles, dragonfly nymphs, water striders, paper wasps, hornets, or tent caterpillars, Charles studied them all. He taught cockroaches to find their way through a maze, proving that they could learn. He triggered moths to beat their wings whenever they heard a whistle, just like Pavlov, a Russian scientist who trained dogs to drool whenever they heard a bell. He frightened doodlebugs, antlions, which made them lie still. With a magnifying glass, he studied how they moved and trapped their food, how antlions grew, and everything else that antlions do. Charles learned so much that he became the world's first doodlebug expert. Patiently, he watched 1,000 caterpillars crawl, slowly, slowly, up a vertical maze, learning slowly, slowly that caterpillars find their way by trial and error. Charles Henry Turner was indefatigable. His mazes, spider jars, paper circles, and cardboard stages toppled old ideas about insects. He never stopped inventing new ways to study the smallest creatures, searching for new ideas or asking questions. Questions that itched like mosquito bites, questions that tickled like spider webs, questions that only a good experiment could shoo away. As hardworking as the ants and bees he studied, Charles published over 50 scientific papers on everything from bird brains and the bathroom habits of cockroaches to blind crayfish and the growth of grape leaves. He was a pioneer scientist of animal behavior, an internationally admired entomologist, and one of the leading African American scientists of his time. But even though he was a respected scientist, Charles faced racial prejudice. He lived in the South where African Americans had to attend separate schools and where they could rarely vote. He lived in St. Louis during the terrible East St. Louis riot when hateful mobs killed more than 100 African Americans and burned their neighborhoods. Yet despite this prejudice, Charles wove himself into his community. He raised money for poor families and led a settlement house that sold lunches to hungry children for only a penny. He worked with white and black people to make St. Louis a better place for everybody. Biology, the study of plants and animals, gave Charles hope. He wrote that biology could help people see the connections among all living things. Biology taught us to think less about ourselves and more about others. Charles Henry Turner, the boy who never stopped asking questions, grew into the tireless reader who owned 1,000 books. He was good friends and always willing to help other scientists by offering ideas, helping with their research, or sharing his equipment. If my micro camera proves a success, I shall be glad for you to use it whenever you desire. Charles Henry Turner, the boy who never stopped asking questions, grew into the determined biologist who would stay up all night to watch a spider or spend all day observing wasps beside a railroad track. I shall take my camera along and get a few photographs. His curiosity danced from experiment to experiment. It moved in Turner's circles, in Tourment de Turner, always exploring, always reaching to discover new ideas. Charles Henry Turner, the boy whose teacher urged him to go find out, grew into a teacher himself, a devoted scholar who taught students to look closely, to find the webs that connect us all, and just as he did, to fill the world with questions, questions, questions. So that's the end. There's an author's note. This is a picture of Professor Turner, and this is another picture, and it said, "There is much evidence that the responses of moths to stimuli are expressions of emotion. The fact that an insect does not respond to a sound is no sign that it does not hear it. The response depends upon whether or not the sound has a life significance." Okay, so now that we've read the book, what you're going to do is you're going to go ahead and get out your printable. This should have been in your garden kit. And you're going to go ahead and get out your bug jar. Okay, so now that we've read the book and we've learned all about Charles Henry Turner, you're going to go find your own mini beast. And I say mini beast because we don't like to call them bugs, because a spider, okay, is a spider a bug, an insect? No, a spider is an arachnid. Okay, this is my spider. You can see him kind of moving around in here. Okay, so that's my spider. Then what else did I find? I found a little ladybug. I'm not sure if you can really see it, but oh, see it? He's moving around, walking around the inside. Okay, and these bug jars have little teeny holes in the lid that allow some air in. So your mini beast is going to be fine in your bug jar. Okay, you can see him walking all around. These lids are also, if you kind of push them down hard, they're going to stay on even if you tilt it upside down, which we have some in the garden room that the lids tend to fall off. These ones won't do that, so you don't need to worry quite as much. And what's nice is you can see the underside. So see, the ladybug was walking upside down, so you can see underneath that shell, which is very cool. Okay, but what you're going to do is you're going to find a mini beast. And once you find your mini beast, you're going to go ahead and you are going to think and observe your mini beast and write down a question that you have. So when I first grabbed my little buddy here, my spider out of my web, okay, he played dead. He curled up in a ball, and I thought maybe I had hurt him. And I was sad because I don't want to hurt my bug or my arachnid. Okay? But then I realized that he was just playing dead because he quickly came back to life and started going all over the jar. So my question might be, why does my spider play dead? Or why does my spider curl up when I get near him? Okay? And maybe an experiment to answer that question is, does he curl up necessarily if I gently tap? No. Okay? So it's not a sound thing. If I use my voice and I use my strong voice and I vibrate the container, does it seem like he's moving very much? No. So is it sound necessarily that causes him to curl up and play dead? I wouldn't think so, right? Maybe it's when the habitat changes or the web changes, right? Because he was in his web, that was why he was easy to get. I kind of destroyed the web when I put the bug jar through it to get this little guy. Okay, maybe he senses if the web has too much vibration that it might be a predator and he curls up and plays dead. Okay, so an experiment I have to answer that could be I touched a spider web to see what happened to the spider. Did he curl up, right? And pretend that he was dead. Okay, that could be an experiment to answer it. If you're going ahead and playing with your ladybug, okay, maybe I want to figure out, can ladybugs walk upside down, right? And so an experiment to answer that would be to put them in a jar that has a lid and see if they walk across the top of the jar, right? Can they walk upside down? And we did just kind of see him. He was walking upside down a little while ago. This is where, just like Charles Henry Turner is really patient, you're going to need to be patient too. And you're going to need to watch your bug. And as you watch him, you're going to say, please, 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 see if you can come across, come upside down. Or maybe if he's on the bottom, look, oh, I flipped him upside down. So I know that he can walk upside down now. How? An experiment to answer it was, I flipped my jar upside down and saw if he stayed on there. Okay? Maybe you have, in the last class, I had this one's empty, but I had a snail in here, right? And maybe one of my questions is, do snails eat lettuce or strawberries? Do snails prefer, right? And so an experiment to answer that question would be, I'm going to put my snail on a plate with a piece of lettuce and a piece of strawberry and see which one the snail eats, okay, where the snail goes. That's a great experiment to answer them. Okay, so it is totally up to you. You're going to figure out your own experiment, and you're going to really watch your bug. Good scientists, what they do is they repeat the experiment lots of times, because just like if you and I walked up and somebody offered us, maybe they offered you a strawberry and me an apple, right? I might prefer the strawberry and you might prefer the apple, but somebody else might prefer the strawberry. So sometimes it might just depend on some other factors as well. So they repeat it enough times to make sure that it's not just a one-off, that it's a pattern. It's true of most things, okay? But so what you're going to do is you're just going to watch. Maybe I have a question, can a spider make a web in a container this small, right? So I'm going to watch him and see. It looks like he's almost trying to make something right now as he's going along in that jar, okay? We can see if we can even get closer. Let's see. Okay, and so these bug jars, again, your insects should be able to last for a couple hours for sure, easily in them. Okay, you can see my spider is definitely working on something. And you can definitely see my ladybug is now walking upside down in my container all over the place. All right, what you don't want to do is you don't want to design an experiment to hurt your mini beast. All right, because at the end of the day, you want to be able to release them. I know a couple students have asked, well, can I make an arachnarium like was in the book, right? If you want to take a mason jar and make something for your mini beast to live in, that's great. Just make sure your parents know and they're okay with it. So that's everything for this week. Hope you guys enjoyed the garden lesson, and we will see you next week for the next garden lesson, all right? So have fun getting out there, see what you can find, whether it's a little ladybug or a nice little spider, okay? Have fun getting outside.